Morning. So today I want us to dive a bit deeper into the digestive system. And in anatomy lecture, you already discussed the organs and accessory organs that are part of the system. And in our warm-up activity earlier, you were able to identify the basic functions associated with the organs. So when I'm saying diving deeper into the digestive system, I just want you to understand how food is broken down and where is it broken down, and actually also how much is broken down. Now, if we take a big bite of a breakfast burrito as I had this morning, our digestive system will break it down into its components or nutrients, which means it breaks it down into carbohydrates, proteins, lipids, also DNA and RNA, and of course, vitamins and minerals. If we look closer now at the carbohydrates, and as you may recall, the smallest unit of carbohydrates is the monosaccharide or simple sugar. And if you, as a team, now could please look at the cards in front of you and pick out those cards which show monosaccharides. So great, you identified glucose, fructose, and galactose, and some of you also identified ribose and deoxyribose as monosaccharides. Now, ribose and deoxyribose are also monosaccharides, but we will talk about them later in the context of DNA and RNA. Now, disaccharides, as the prefix gives away, these disaccharides are composed of two monosaccharides. So I would like your team now choose the three cards that show those disaccharides. There they are. That's maltose, sucrose, and lactose. The lactose, the disaccharide of intolerance. So if we put more than two units together in some way or the other, we end up with polysaccharide. And as you may remember, starch, glycogen, and cellulose are representatives of this group of polysaccharides. And we have our cellulose, we find in plants. Glycogen is how we store our glucose in our body, in the liver. And starch is, for example, in our food, in our flour, when we bake bread, that has a lot of starch. So I have one more team task in this section, and I want you to pick the two cards that represents the two simple sugars that make up the disaccharide maltose, and then the same thing I want you to do with sucrose and then with maltose. All right, well done. So we have maltose, which consists of two glucose units. Then we have sucrose made up of fructose and glucose, which is our table sugar. And then we have lactose that is made of galactose and glucose. And now that we have resolved all the confusion, let's have a piece of bread right here. That's yummy. The flour in this bread contains, besides proteins, it contains a lot of starch. And as we know, starch is a polysaccharide. Now, starch is a long string of glucose units connected to each other, and these strings have a name. It is called amylose. Some of those strings are more branched, and then we call them amylopectin. And both of them, amylose and amylopectin, can be broken down by an enzyme we call amylase. So amylose, amylase, to get them not confused, you have to remember the suffix of the word. So sugars have the suffix -ose, whereas enzymes have the suffix -ase. Now, amylase is produced in 3 areas of our body. And when we chew our bread, salivary amylase in our mouth is released by 3 salivary glands. And if you chew long enough, you may even detect the sweet taste of your bread. But our salivary amylase does not do a very good job in breaking down the starch. So our food particles mixed with the saliva, what we call the bolus, now travels down the esophagus into the stomach and then into the small intestine. And here the starch particles reach the duodenum, the first section of the small intestines, where our pancreatic amylase comes into action. It further breaks down the starch mainly into the disaccharide maltose, which consists of 2 glucose units. And finally, we have our glucoamylase, which is a brush border enzyme, and that is released by small cells in the small intestines. So it breaks up the bond between the 2 glucoses, which means we end up with the simple sugar glucose. Now, our enterocytes, which are the simple columnar cells in our intestines, are able to absorb our glucose with the help of a glucose-sodium cotransporter, and then glucose travels through the cell to the other side where it accesses into a capillary. And now that glucose has reached our blood system, it can be transported wherever it's needed or stored as glycogen in the liver. And the sodium is pumped out of the cell by a sodium-potassium pump, and we're getting back to normal. So sodium out of the cell, potassium inside the cell. So our first substance, glucose, has reached its destination. Now, if you look at this list, we have as our source carbohydrates, it shows our monosaccharides, the substances which can be absorbed, and we have lactose and fructose, which go a similar way, like glucose and as glucose. Now, fructose is been metabolized in the liver and it turns into glucose. But if you are interested, this can be one of your research projects to look a little bit into the lactose, galactose digestion or the sucrose, fructose digestion. If you want to do that, come talk to me after class. Now, before we continue and look at the proteins, fats, and the nucleic acids, I would like you to answer the first 2 questions of your handout. And here they are. So I give you time for that. But moving on, since we already talked about the monosaccharides, ribose, and deoxyribose, let's see how they fit into the picture. Ribose is part of the RNA molecule, the ribonucleic acid, and deoxyribose is part of the DNA, the deoxyribonucleic acid. The smallest unit of the DNA and RNA is the nucleotide. And a nucleotide is a simple sugar plus a phosphate group and plus the base of the that contains a nitrogen. So if we connect a bunch of nucleotides, we get the helix shape for RNA and the double helix for the DNA. Now, all animal cells and plants have DNA and RNA. That means when we eat meat or a salad, we also digest the DNA and the RNA of these animals and plants. Now, again, we have enzymes that take care of the breakdown of it. And so we cut the DNA and the RNA with DNase, which is produced in the parotid salivary gland, and in the pancreas, whereas the RNase is only produced in the pancreas. So DNase and RNase cut down the DNA and the RNA respectively into single nucleotides. But we cannot digest nucleotides, so they cannot be absorbed by the cells in our intestines. So we have to cut them down even more. So we have some brush border enzymes that come to help. And here we have the phosphatase, which cuts off the phosphate from the nucleotide, and the nucleosidase, which cuts off the base on the other side. Now we are left with just like these parts here, and we can look in our little table that we see on the nucleic acid. We have the pentose sugar, the phosphates, and the bases, which are the substances that can be actually absorbed by our cells and go into our system. And here we are. Now, what I would like you to do, look at the assignment number 3 and create the pathway of the DNA with the given information you have there. And we'll give it a big nice